Hello, welcome to Arya Mate. I am Arya, I'll teach you Mate. So today let's start with a very interesting topic which is mitochondrial myopathy. Actually, this is a very rare disease. The reason is because there is something known as heteroplasmy. What is heteroplasmy? See, you may have some abnormal mitochondria in a cell, okay? However, there are normal mitochondria as well. So it doesn't mean that every cell having abnormal mitochondria would express the disease. In some cell, maybe all normal mitochondria. In some cell, maybe all abnormal mitochondria. So that is considered to be heteroplasmy. That means every cells may be different, and in a same cell, there may be different mitochondria. Some may be normal, some may be abnormal. Okay, and there is something known as threshold effect as well. In the threshold effect, the most important thing for us to remember is in a normal individual, mitochondria are normal. Even in the abnormal individual where the mitochondria is abnormal, in that case as well, the symptoms may not appear until and unless the number of abnormal mitochondria is above a certain critical level. Like I mentioned, in a cell there may be normal and there may be abnormal. Some may be normal, some may be abnormal. And the normal mitochondria carry out the functions really well until and unless during exercise or during disease or in any condition, the function of the cell must increase and the cell cannot cope up. So that is considered to be the threshold effect. So mitochondrial myopathy does so threshold effect as well. Now, one of the thing about this disease is it is maternally inherited. Why? Because ovum is the source of mitochondria. In sperm, mitochondria does not survive. It's degenerated. And that's important in order to pierce the ovum, in order to travel such a long period of time. It cannot carry a lot of organelles. It does make sense, isn't it? So, ovum, that means maternal inheritance. It is a disease of maternal inheritance. But it doesn't mean that if the mother is infected, the child would also be infected. Why? Because in the ovum, some healthy mitochondria may also go. Or let's say all the mitochondria may also be healthy. Or all the mitochondria may be abnormal. See, this is a disease of heteroplasmy. This is a disease of threshold. So, you won't be finding these diseases in your clinical time a lot. It's very rare condition. However, we have to study because even rare condition, our patient would die. Our patient would have problem. So we have to know something about it, isn't it? Even one life matter as a doctor. It's a ethic. Okay, so let's go. So what are the manifestations? Actually, the mitochondria myopathy. That means mostly we are talking about the manifestation in the muscles. Why? Because muscles has higher requirement of energy. Not only muscles, even nerves also have higher requirement of energy. So most of the manifestation of the disease is in the muscle or the nerve. So let's say this is an individual. So what are the problems this individual will have if the individual got mitochondrial myopathy? Everything starts with its brain. In the brain, they may get dementia, ataxia, seizures, vision loss, means the area that is responsible for the vision is lost, hearing loss, that means the area that is responsible for hearing is lost. Yes. Then we have eye. In the eye, they get cataract. They can get retinopathy. In the heart, they get a lot of cardiopathy, especially the one that is related with the conduction, especially cardiac conduction deficit. Kidney as well. In kidney, the person would get Fanconi syndrome or nephritis. Even in the liver, they would get hepatic steatosis. That means a lot of organs can be affected. Obviously, we cannot forget the muscles. Muscles myopathy would be there. There would be tremors. There would be hypertonia, hypotonia. Anything could be there. Convulsions. muscle-related convulsions, nerve-related convulsions. There can be neuropathy, a lot of things like that. Okay, so we'll be dealing about this in detail when we go to the individual disease. So let's go to the individual disease. First, we have Kern's sires. So when I remember this disease, I remember from a mythology of Krishna and Karns. Karns was a demon, isn't it? Was he a demon? No, he was actually maternal uncle of Krishna. Let's not go to that. However, he was a very cruel king and he has a sire. Sire means a poet. So how that poet looked like? The poet was quite of a short stature. Okay, quite of a short stature. His age was less than 20 years. So this disease manifestation is mostly less than 20 years. The manifestation of disease include the less height. And he had an eye cough. Why? Because he had a vision lost. And one of the eye had ptosis. He used to drink a lot of alcohol as well, and because of that reason he had ataxia and muscle weakness. He had dementia and he had hearing loss as well. So if you remember this much, you would definitely remember Kern's sires. Now let's come to another disease known as Leigh syndrome. In Leigh syndrome, it's normally a disease of childhood, especially infant. Here also we have ataxia, dementia, seizures, developmental deficits, weight loss, and a similar type of symptoms. We have Leber optic neuropathy. In Leber optic neuropathy, we'll be observing optic neuropathy, especially retinal vision loss. That is important. We have MELAS. What is MELAS? It is considered to be mitochondria encephalopathy. See, this is mitochondria encephalopathy, lactic acidosis, and stroke-like syndrome. Actually, here in this leg also we get lactic acidosis. You know why we get lactic acidosis? See, mitochondria is normally responsible for producing those genes that regulate our electron transport chain. So as the electron transport chain is affected, that means the pyruvate has to be sent to the lactic acid formation. It cannot be utilized in the electron transport chain, right? So that's important. And one thing I forgot to mention last time, all the activities of mitochondria is not regulated by mitochondria. Some of them are even produced by DNA. That means DNA also regulates the protein synthesis of our mitochondria. Only about 13 proteins are synthesized by mitochondria, and that's the reason as well for the heteroplasmy and the rarity of the disease. Encephalopathy means the problem is with the brain, which is obviously going to manifest as a seizures, dementia, ataxias, right? Lactic acidosis, stroke-like syndrome. That means patient get a lot of transient ischemic attack and stroke as well, and obviously the patient would get seizures. And let's see, encephalopathy, lactic acidosis, yes, from the name itself we can understand. Another is MARF. It is myoclonic, MARF, not MARF, okay, myoclonic epilepsy, and this is ragged red fiber. Actually, these are fiber-like structure seen in the cell in the microscopy, which denotes a lot of abnormal mitochondria. So obviously, you will be observing ragged red fiber in this disease. Myoclonic means you will be observing myoclonic condition, hypertonia, convulsions, right? And epilepsy means a lot of seizures and convulsions. Even lactic acidosis is also common. Ataxia, dementia are also very common in this disease. Now let's talk about nerve. I think it's more of a nerve. Okay. In nerve we have got neuropathy. Yes, you get a lot of neuropathy. Ataxia, retinitis pigmentosa. It is NARP. Okay, now in this disease you can see there can be neuropathy, ataxia and retinitis pigmentosa, which can lead to the vision loss. Obviously lactic acidosis are also there. So yeah, most of the symptoms that you observe in this disease are very similar to that of the name. So that was all about the mitochondrial disease. There was one more disease actually, MDS, which is mitochondrial DNA deficit syndrome. Okay, so that is also there, and the similar manifestation like we have lactic acidosis and all those conditions. Okay, so that was all from the type of diseases. But now let's go to detection. How do you diagnose? Obviously, diagnosing this disease just based on the clinical features is very difficult, because this kind of clinical features can be observed in a lot of conditions, and this disease is very rare. So what do? We take muscle biopsy. Muscle biopsy is considered to be the gold standard test of our mitochondrial myopathy. In muscle biopsy, we do histochemistry first. In histochemistry, we see different enzymes, whether they are present or not, and our major target is cytochrome C oxidase. So we check whether this cytochrome C oxidase is working or not. Then we have immunohistochemistry. We see the presence and absence of different types of proteins. And obviously our protein target would be the proteins that are produced by mitochondria. We'll do some biochemistry as well. In biochemistry, we use polarograph. And using this polarograph, actually we can detect whether there is presence of oxygen or not. And we can even see through the electron microscope presence of some abnormal mitochondria. Now another is enzymes. In enzymes, best one is LDH, lactic acid dehydrogenase. It would increase in the context of mitochondrial myopathy. Similarly, keratin kinase as well increases. It also increases in MI. This both can increase in MI, but you have to be careful whether that's a hemoglobin. Lactic acid dehydrogenase can also be present in hemolysis. So every time you get lactic acid dehydrogenase increment doesn't mean that person has mitochondrial myopathy. It could be a simple hemolysis as well. And this keratin kinase actually increases in the context of rhabdomyolysis. Rhabdomyolysis means the degeneration of our skeletal muscles. And skeletal muscles do have a lot of keratin kinase, because it acts as an immediate source of energy after ATP has been utilized. Even in the anaphylaxis as well, we can have keratin kinase increment. Okay, however, we have to be careful. We have to ask the history, we have to ask the family about the generations and everything, and we have to narrow it down. Okay, we can even see lactate pyruvate ratio. However, it is not very much let's say good in order to find out the mitochondrial myopathy. Okay, similarly, we can see some of the functions of the liver, because it can affect the liver. So you can check the serum transaminases, you can see albumins, and you can even see the bilirubins, okay, in order to see whether your liver is functioning well or not. And there is one enzyme component which is known as 3-methyl gluticonic acid, which is actually formed by degradation, or let's say metabolism, of amino acid lysine. So you can see its label in the blood and the urine as well. So this is also a very interesting method of diagnosing the disease. They are not directly used for the diagnosis, however they can help a lot in order to narrow down the disease. So that was all from the mitochondrial myopathy. If you love it, give a big thumb up. If you do not like it, write down what do you want me to improve. That was all from my side. Stay curious, stay learning. Bye bye.